Okay, so welcome to this session for the 2021 Biodiversity Genomics Conference, which is going to showcase the work that the BAT1K consortium is doing and the work that the members of BAT1K have been doing with the genomes generated by the project. This actually marks the anniversary of the first satellite meeting of the BAT1K community that took place last year at the Biodiversity Genomics Conference. And because it was a satellite meeting about the BAT1K project, we decided to call it our Batalite meeting. This took off, it was very popular and has now become a regular meeting that's been open to the whole community. We have monthly or bi-monthly Batalite meetings. And I just wanted to mention that we actually have a Batalite on the 5th of October, so coming up quite soon, 5th of October at 18:30 European time, that's going to focus on bat conservation and how BAT1K genomes can contribute to conservation efforts. BAT1K is a very open consortium. We're currently more than 300 members strong, and we're always looking to work with anyone interested in bats and bat genomes. So please consider signing up on our website if you're not a member already. And website is bat1k.com. On the website, there's a lot of information about the consortium, about our goals, our progress, and it's also possible via the website to register for our Batalite meetings and join our mailing list. Today, we have 5 speakers from the BAT1K consortium. First, we'll hear from my BAT1K co-director, Emma Teerling, who will give a view on the BAT1K project, the past, the present, and the future. Then we're going to hear how some of our members have used these BAT1K genomes to learn about bat biology. So second up will be Alexis Walid Ahmed on convergent and lineage-specific genomic changes and adaptations to sugar-rich diets in bats. Third, Veronica Lane will talk about how reference genomes are assisting subarctic bat research. And fourth, Ariadna Morales will highlight how BAT1K genomes of potential SARS-CoV-2 reservoir species can provide insights into important immunity changes. Lastly, I'm going to talk about how bats and bat genomes can shed light on vocal learning and human speech and language. For your information, all the talks will be played back to back, and then at the end, we will have an open Q&A and discussion session addressing questions to all the speakers. The Q&A box will be open throughout, so if you have any questions for any of the speakers, please feel free at any time to put your questions in. It would be really helpful if you could also tag the speaker that your question is directed towards. And feel free to also upvote any questions that you want to hear answered. So with that, I'm going to hand over to our first speaker, Emma Teerling.
Good evening. What I want to do is I want to tell you about why we should study bats and why we should look at their genomes. And I want to give you an update on what we as a group called BAT1K have been doing. But first of all, let's talk about these wonderful and fascinating mammals. Well, if you think about it, there are lots and lots of bats. 1 in 5 of every mammal alive on this planet today is a bat. But they're not just extraordinary because of their huge numbers of species. They're extraordinary because bats have a number of unique mammalian adaptations. Bats are the only group of mammals that have achieved the ability of true self-powered flight. Bats are able to uniquely use laryngeal echolocation to orient in complete darkness. Bats are crucial to how our ecosystems function, being major keystone predators of arthropods and also key pollinators in the tropics. But 2 other unique adaptations that bats have can greatly help us advance and improve our future societies. And one comes down to the fact that bats have been shown to be carriers of many different viruses without becoming sick, suggesting they've evolved a unique and tolerant immune system. But also bats are extremely resistant to the aging process. Bats are very small mammals with a very high metabolic rate, but they can live for an extraordinarily long time. These 2 adaptations we're going to focus on. The idea that bats have evolved a unique and tolerant immune response. And also this is directly relevant to the current pandemic we're all slowly getting out of. Bats have the highest diversity of coronaviruses that have been studied so far. This indicates that bats can live with coronaviruses, such as SARS-like coronaviruses, and not get sick. Therefore, studying bats' immune response can allow us to understand how we can better tolerate and live with these viruses. But bats also appear to have evolved these extended health spans. This is a bat that holds the longevity record. It's Myotis brandtii. This individual was caught as an adult, tagged, recaught 41 years later. And what's extraordinary is not necessarily the 41 years of age, but the fact that this bat can live over 40 year ones of age, yet weighing 1/3 that of a lab mouse. So bats should die young given their small size and high metabolic rate, but they seem to have bucked this trend. So therefore, studying bats can allow us to understand how we can age more healthily, and also can find novel ways to fight infectious diseases. But how are we going to do this? Well, to be able to understand the unique molecular adaptations that bats have evolved, we need exquisite genomes. And this is where BAT1K comes in. So what is BAT1K? So we are a global consortium of people, like-minded people, that want to sequence the genome of every single living bat species to chromosome level error-free assembly. And we want to do this because we want to uncover the genomic basis of their unique traits, their extraordinary adaptations, and ultimately we want to drive a global conservation perspective and awareness of bats. So just to remind us all who we are, so myself and Sonia Vernes are the two founding directors of BAT1K. David Ray from Texas Tech, Jean Myers from the Max Planck, Michael Hiller was in the Max Planck, was now in Senckenberg, Germany, Elena Davlos for Stony Brook, are all members of the executive of BAT1K. And really, we came together about 2016, published our white paper in 2018, with the idea that actually we want to sequence these genomes. And how do you sequence 1,400 bat species? Well, we decided to break them up into more manageable tasks. And our first pilot study had 6 key species of bats representing basal diversity, different adaptations, whereby we wanted to see, could we find ways to be able to ship the tissues around the world? Could we sequence them? Could we extract the DNA? What different types of sequencing methods do we need to put together? How should we assemble them? But see, could we sequence exquisite bat genomes? That was our pilot study. We divided up the other phases into family level phase, general level phase, followed by species level phase. And indeed, July 2020, we published our pilot study. And we published the first reference genomes of 6 species of bats, exquisite genomes, where we analyzed them using state-of-the-art molecular phylogenetic methodology. But could we find any evidence of bats' adaptations in these genomes? So could we find, for example, a signature of unique immunity or viral diversity? And the answer is yes. So when we found and looked at these genomes, we found evidence of selection, loss, and gain of innate immune genes and also antiviral genes. For example, in bats, you find that the ApoBec gene family, antiviral gene family, is expanded. You find loss of key genes involved in inflammatory pathways. And this suggests that bats have evolved mechanisms to modulate their innate immune response to allow them to tolerate and live with viruses. We also found a huge diversity of endogenized viruses in bat genomes, giving us evidence that bats are able to survive past viral infections. And also maybe looking at bat genomes sequenced this way can give us an insight into what other viruses they may carry. Could we get an insight into future pandemics? So our pilot study was finished and we moved on to phase 1, our 21 bat families. And at this stage now, what we have is we've sequenced and assembled 17 of the 21 bat families. Remember, they're found throughout the entire globe. We've sourced tissues from every single bat family, so we know where they are, apart from one, which is Mysopoda. We're working with Neil Gemmell in New Zealand to try and see, can we find this unique species? What we've also done is we've sequenced and assembled 9 key rhinolophoid bats, potential reservoirs of SARS-like coronaviruses, to advance research into COVID-19 resistance. Overall, we have 35 bat species that are currently being annotated. But what's really exciting is that of these 35 bat species, 17 of these bat species are at this new VGP 2020 assembly metric, which is different from even what we published last year. As you can see, here is an example in green of our 17 different bat species from right across the bat tree. And indeed, this is 7C Q50C95, which is we can achieve with these hi-fi assemblies. You have a contig N50 greater than 10 MB, a scaffold N50 at a chromosome level, base accuracy at 99.99%, and greater than 95% of our genome is scaffolded into chromosomes. So what are we going to do with all these new genomes? Well, our family is also expanded. We have a new key group of steering committee members involved in looking at genotypes, the phenotypes, looking at conservation, our assembly annotation crew, looking at immunology and pathogens, taxonomy collection permits, basically expanding the group of members who are actively involved in BAT1K. We also have realized that we need to try and source our samples. So what we have done is we've recruited key regional chairs, and you're going to hear stories from some of these. This is Veronica today. They're found throughout the entire world, and that these individuals are responsible for helping us source and identify key species, and for us all working together. BAT1K has always been a bottom-up approach, and we want to bring our entire community together. We have over 315 consortium members. We have a Twitter following of about 1,200 people. What we're also involved in right now is collecting tissues and helping the Darwin Tree of Life project move forward. And our BAT1K members are collecting bat tissues from throughout Ireland and the UK, and also we're expanding this out to include European bats. So I hope I've convinced you of the utility of BAT1K, that we should all be working in sequencing bats, and because what we really need to make any genome consortium work is we need people to join us, to help us find the samples, to help us sequence samples, annotate the samples, analyze the samples, and move everything forward so we can uncover the unique annotations that bats have evolved. Thank you.
Hello, welcome to this talk about amino acid catabolism and sugar-rich diets in bats. I'm Alexi Walid Ahmed, a PhD student at the Center for Translational Biodiversity Genomics in Senckenberg in Frankfurt. So one thing to know about bats is that you can find pretty much all the diets you can think of in them, but paradoxically, all this diversity is concentrated into 2 main families, and almost all other bats are insectivorous. So the first one is the Pteropodidae, and the second are the Phylostomidae, where most of the actual diversity happens, but we will be focusing on the fruit and nectar eaters for our purposes. We also mentioned that we have 2 new long-read-based assemblies for Glossophaga soricina and Carolia spicillata. So such a shift to a diet of this caliber is kind of difficult to explain if you take into account that nectarivorous, the insectivorous bat, will have a diet that is very rich in protein and lipids, and here you have the exact opposite. It's poor in both of them and very rich in sugars. So kind of we're trying to probe the genomic basis for this dietary shift. To do this, we're using a tool called TOGA, which tries to map inactivating mutations onto all the genome-wide, onto all the possible transcripts. So it looks for splice site mutations, premature stop codons, frame shifts, anything that would prevent this gene from being expressed anymore. If we look for the genome enrichment analysis in the gene losses that are specific to the Pteropodidae and the Phylostomidae and that are never present in any other bat, we see that we have many things of interest, especially in the Pteropodidae, lots of stuff related to lipids and so on, but also in the Phyllostomida with SLC-mediated transmembrane transport, which would probably tie in with water balance. But for the purposes of our talk, we will be focusing specifically on tryptophan catabolism. So let's introduce the Canurian pathway. The main point of it is to take tryptophan and to create NAD+ from it in the liver. But it's also a main sink in circulating tryptophan. If you inhibit it, you get a 9-fold increase in the circulating tryptophan. It also has a bunch of multiple minor branching pathways that do other stuff. So if we split it into 3 steps, the first is the entry into the pathway. Naturally, it's going to be rate limiting. And we have 3 enzymes that do that. TDO is expressed in the liver, it's the main one. IDO2 is kind of ubiquitous. And then you have IDO1, which is expressed specifically in the immune cells, placenta. There's a rationale behind that is that basically they deplete locally the amount of tryptophan you have available, and that starves the pathogens. So if we look at the gene losses, we find that IDO1, the immune one, is never lost. However, in contrast, the ubiquitous IDO2 is lost in all the pteropodids and convergently with Tornera hondurensis. And then you have TDO, the main one, is lost in the Rosettis genus. I can't resist showing you what's actually what TOGA outputs. So as you can see here in IDO2 for Rosettis and Pseudophan, all pteropodids, you can see this common premature stop codon and this common frame shift. This indicates that this happened much, ancestrally, probably broke the gene. And then all the rest is accumulated later because the gene evolves neutrally. Now, if we focus on the main pathway, we find that it is lost convergently twice in the Rosettis genus and in Glossophaga. I grab your attention to Kainu, which is lost in none of them. This is because the gene is pleiotropic, so you could not actually use it. And finally, there is one side reaction that was lost convergently multiple times. So ACMSD is responsible for making picolinic acid, and ALDH8A1 takes a pathway into glycolysis. And we find that the second one is lost in all the pteropodids and convergently in one phylostomid lineage, the Stornera and Artibius. In contrast, ACMSD has been lost at least 4 times independently, twice in the phylostomids and at least twice in the Rosettis. So if we take a kind of summary view of this, all the possible ways in which you could affect this pathway so that it does not do its job can be found convergently across these 2 lineages. So it seems to be following precisely this dietary shift. So the hypothesis, what's the hypothesis for this? The idea is that in insectivorous bats, they have plenty of amino acids because from their food intake. And so when they undergo some fasting, they can, without any problems, deplete some of their proteins to get some amino acids, which they can catabolize for energy. And then in the next feedings, they will replenish all their reserves. No big deal. However, in sugar-eating bats, they have a smaller pool to begin with. And this will be especially a problem for essential amino acids that are rare, such as tryptophan, because then you have catabolisms that can deplete it, and you have no way of actually getting it back into the proteins. So there are 2 ways you could try to address that. The first one is inhibiting catabolism. This is what we saw with canurinin, but we also thought of a different way this could happen, which is just incorporate less of it into your proteins, start being parsimonious with it. So to do so, we basically looked at all the genes in the bats, and we looked at the relative frequency in tryptophan. As you can see, there is a net, very obvious shift downwards for pteropodidae. They are depleted. Their proteins are depleted in tryptophan. In Phylostomidae, however, there are more conflicting results. We're still in the process of understanding them. For example, you can see that the one that has a full pathway loss is pretty low for a phylostomid, but pretty average for most bats. So this can give us multiple hypotheses, and we need to test them to actually look at the level of precise proteins. Because, for example, maybe they are just more, they have adapted more effectively to it and have just depleted specific key proteins that are expressed in the liver or more expressed to have this amino acid shift. However, the overall conclusions are pretty solid. We have found numerous convergent gene losses targeting the kynurenine pathway. We have additional evidence for these losses co-occurring with genome-wide shifts in amino acid composition. And we hypothesize that this is due to their protein-poor diet leading to a depletion in tryptophan and a need to conserve it. Okay, I want to acknowledge the Hille Lab, especially those who have worked on this tool. I want to thank my thesis advisory committee for their very useful comments. And I want to thank the MPI of Cell Biology and Genetics in Dresden, from which most of the sequencing facilities and sequencing expertise has come for those two genomes. Thank you very much. I hope I interested you and convinced you of the thing. Bye.
I'll be talking about our reference genome projects here in the BAT-Lab Finland, in the Finnish Museum of Natural History in Helsinki. In our research, we are really interested on the subarctic area, which is in the northern hemisphere, like right below the Arctic Circle. And especially we are interested on the Nordic countries, but also in bat research in Russia and also northern Japan. And we are doing research on, especially on two species, and we're also concentrating on their reference genomes. And the species are, the first one is the northern bat, Eptesicus nilssoni, which is a common bat species in the north. You can find it here in southern Finland, all the way to the northern parts of Finland and also Norway. It's really a generalist species, and you can find in different habitat types, also in the cities. And it's not a migratory species, so it's more sedentary species. And we have done genome assemble with the help of Dovetail Genomics. We had reads for the genome assembly, we had reads from BacBio and OmniSeq, and that's now done. So currently we are doing RNA sequencing for the annotation. And hopefully later this year we have this genome available for our research. And the other species is the Nathusius pipistrelle, Pipistrellus nathusi, which is a newcomer in the north. In the recent, maybe like 10 years, it has become more and more common here in Finland. It's a really common species in the Central Europe, so it's spreading to north, and it's a migratory species. So currently in autumn, it's actually migrating from Finland to Europe, and it will be back here in spring. With this species, we have now a draft genome based on BacBio sequences, and we will continue this genome project with BAT1K and Sanger Institute in order to make a proper reference genome for this species as well. So why are we interested on these two species? So with the northern bat, we are really interested on the genetic population structure throughout its range from Europe to northern Japan, in order to understand adaptation to different environments and also to the northern climate, but also understand its population decline. So we have seen here in the Nordic countries that the northern bat populations have been going down. And we don't really know why. So it could be climate change or habitat changes in general, or maybe competition with the pipistrelle bats. So in order to understand that better, we need this kind of study. And I'm especially interested in a certain kind of adaptation, which is adaptation to urban environments. So as I said, it's a common bat in the cities. You can see it here in Finland in many of our cities and flying under the city lights. And I would like to find out what are the genetic changes or epigenetic changes that are related to urban living in the northern bats. With the Nathusius pipistrelle, we are really interested on the population-specific migration patterns, and that linking with the pathogen dynamics. So here we are looking at the genetic population structure, also temporal variation, and also looking at the microbiomes from the fecal samples. We have samples along the main migratory route, also from different years in order to cover the temporal variation. And currently we have samples from Finland, Latvia, Russia, Poland, Belarus, and Ukraine. But we are interested to get more samples. And so if you have tissue or fecal samples available, we are happy to collaborate with you. So in the end, we would like to identify potential pathogen source populations in these projects. So these two projects we have just started. So we are currently building the reference genomes, and so unfortunately I don't have any results yet, but the genomes are already almost done. So the next phase is to answer these questions and use these genomes. So what we are going to do in the next couple of months, we will, with the northern bat, we will do whole genome sequencing in order to get SNPs and larger structural variation to find which of the genes or genomic regions are related to adaptation in the whole range and also related to urban living. And related with the urbanization, I will also do RNA sequencing and bisulfite sequencing to get the RNA expression and methylation levels for the epigenetic part, again to find which genes are related to urban living. With the Nathusius bat, we are doing rat sequencing in order to get SNPs, and also we are sequencing the fecal samples in order to get the pathogen distribution. And then we're trying to find if these patterns are linked. So hopefully in the coming years, I will have some cool results related to these projects. There are many people and institutes to thank. So first is Thomas Lilly from the Bat Lab Finland, and Emma Teeling from Bat1K, and Steve Patterson from the University of Liverpool. Aleksi Lehikonen and Arto Pulliainen are part of the Nathusius project, and Dovetail Genomics helped us with the northern bat genome, and Center for Genomics Research from the University of Liverpool did the Nathusius sequencing. And I would like to also thank you for listening. Thank you.
Hello, my name is Arieta Morales. I am a postdoctoral researcher at the Center for Translational Biodiversity Genomics, or TBG, in Frankfurt, Germany. And today I'm going to be talking about how genomes of potential SARS-CoV-2 reservoir bats provide insights into immune system changes. This project is part of the Bat1K, and I would like to acknowledge the diverse team of scientists contributing. Coronaviruses were discovered 90 years ago, and those that infect humans have been around for at least 60 years. They are usually associated with mild respiratory infections. In recent decades, viruses such as SARS and MERS have emerged among human populations, and we know that they can have devastating consequences, such as the current pandemic. Bats are suspected reservoirs for many viruses, including SARS-CoV-2. Specifically, bats from the families Hypocideridae and Rhinolophidae are among the main suspected reservoirs, and these species are usually found in Europe, Asia, and Africa. Bats possess a special immune system in which they dampen uncontrolled immune responses and limit cell damage. And these adaptations allows them to live with viruses that can be lethal to other mammals. Therefore, in BAT1K, we aim to understand how bats have co-evolved with coronaviruses. And to answer this, we have generated high-quality chromosome-level genomes of species that are potential reservoirs for SARS-CoV-2. We use technologies such as PacBio, HiFi, and HiC to generate 7 new genomes of the species that I'm listing here. And the viruses found in some of these species are closely related to the strains originally found in Wuhan, China. These genomes improve in quality upon any bat assembly previously published. And these improvements can be shown in terms of contiguity, completeness, and base accuracy. For example, in this figure, I'm showing in the x-axis the percentage of contigs which specific lengths in base pairs that are shown in the y-axis. So for example, this is the N50. And in red, I'm showing the new genomes, which N50s are higher than those genomes previously published, including the best genome, which I'm highlighting in light blue. When these contigs are assembled into scaffolds, these stats are even better, with a maximum N50 of 184 million base pairs. We also measure the completeness, meaning how many genes were missing among a set of 4,000 genes that are found in most mammals. And this is an example of a species in which we were missing 149 genes. But with the new assemblies, which are highlighted here in yellow, the number of missing genes is very low. We use these genomes for a genomic screening, but to be sure that the genomic signatures found in Hyposideridae and Rhinolophidae are specific or unique to these lineages, we also included mammals representing the main taxonomic groups. In total, we have 64 genomes, and all these genomes were annotated or re-annotated using a method called TOGA that is being developed in the Hiller lab, and it is based on gene predictions. We also perform such phylogenetic analysis and divergence times, and we screen for relevant gene changes, such as gene losses, meaning mutations which may have led to loss of function, and screenings for selection and expansion or contraction of gene families. Okay, what have we learned so far from these genomes? We have identified 29 genes that are either lost or under selection and have some relevance for the life cycle of SARS-CoV-2. But to explain why these genes are relevant, first, let's revise briefly the life cycle of this virus. So here we have a cell with a virus where the spike protein is going to interact and attach with cellular receptors to enter the cell and then encode genomic RNA. Immediately after, the replication complex is going to be formed to synthesize new RNAs, and some of these RNAs are going to be translated into structural and accessory proteins that then are going to be used to assemble the virion and then release it by exocytosis. What we found is that the 29 genes are mainly targeting RNA synthesis, translation, and assembly of coronavirus. And an important phylogenetic pattern that we've noticed is that ancestral genes in Rhinolophidae and Hyposideridae may inhibit mainly viral RNA replication. For example, in this phylogeny, I am highlighting ancestral nodes within these families, and the genes under selection in these nodes are mainly linked to synthesis of RNA. And another important pattern is that species may have convergently evolved mechanisms that may inhibit translation of structural viral proteins and virion assembly. Which I mean with this is that species-specific genes may be targeting the same proteins. For example, in this tree, I have different species highlighted with different colors, and different genes are interacting with the same structural proteins of SARS-CoV-2. These results are very preliminary, but for now, we can say that within Hypocyderidae and Rhinolophidae, ancestral lineages evolved adaptations to inhibit viral replication, and then species-specific adaptations may inhibit translation of structural proteins and virion assembly for novel viruses, such as SARS-CoV-2. And we have a list that is an important foundation for experimental validation with these genes. With that, I would like to thank you for your attention. I will be happy to answer questions either at the end of the session, or feel free to send me an email. Thank you.
My name's Sonia Vance. My lab studies what bats and bat genomes can tell us about vocal learning and human speech and language. The reason we study vocal learning is because it's an essential component of human spoken language. It's exactly what we do as children when we learn to speak by listening to the sounds in our environment. But outside of humans, vocal learning is very rare in other animals. Most of the animals we know about that are vocal learners are birds. And many mammals don't learn new vocalizations. In fact, most of the mammals that do learn vocalizations, like seals, dolphins, and elephants, are not very tractable for neurogenetic studies. And so that's why we study bats. These are mammalian vocal learners that are small, highly social, and can be studied at a neurogenetic level. And we can keep them and breed them in captive colonies for those studies. So together with the Bat1K project, we've been generating reference genomes for a number of bats, including vocal learning bats. And thanks to the work of the other genome projects, we are now gaining access to vocal learning genomes for a range of other species, both mammalian and bird. So what can we do with these reference quality genomes? Well, on one hand, given the diversity of species that showed vocal learning, we can use these different species for cross-species phylogenetic analyses to try and really understand what is it that makes a vocal learning genome special. On the other hand, we can look at individual species and perform in-depth studies to understand what those genes do in the context of a vocal learning animal. And I'm going to give one example from each of these types of studies today. First, with the cross-species phylogenetic analyses, I'd like to show some data that we published last year, where we were looking for bat-specific changes in the genome. We found a number of different changes, but one of which was in not a gene, but a regulatory molecule known as a microRNA. This microRNA is known as MIR337, and here you can see the sequence of the human 337. It's a small molecule, only 22 nucleotides long, and it's highly conserved over evolution. However, bats had two specific changes that were found in all bats, and this was really unusual. In fact, we didn't see these two changes in any of the other species that we interrogated. So we've got these bat-specific changes in a regulatory molecule that we predicted would have consequences for gene expression. And we're able to test that by taking it into cellular assays to look at the consequences of the bat-specific changes compared to the human sequence. When we look at these consequences in a cellular assay, we can see that the bat version of MIR337 is only able to target the bat-specific sequences, but not the human-specific sequences. And when we took this more broadly and predicted the consequences across the genome, we could see that there were changes in the type of biological processes that were regulated by the human MIR337 and the bat MIR337. In particular, we saw that rhythmic processes like circadian rhythms, developmental and behavioral processes, and synaptic transmission were present in the bat categories, but not in human, suggesting that the bat MIR337 has expanded its functionality over evolution. Now turning to the single species studies, I'm going to show you work we're doing with this species of bat, Phylostomus discolor. Here, our goal is to understand the role of individual genes and molecular pathways, as well as influences on the brains of these animals for vocal learning behavior. And we've been doing this by generating transgenic bats. We focus in particular on a gene known as FOXP2, which is essential for normal development of speech and language in humans. Children who have mutations in FOXP2 have a severe form of speech and language disorder. They really struggle to learn how to speak. So we know this gene is important for human vocal learning and speech development, and we want to be able to study it in the brain of an animal that is also a vocal learner. So we've been generating viral constructs that will manipulate the expression of FOXP2 in a living bat brain. One of those constructs that I'm showing you today is what we call our knock-in construct. It's a virus that carries the full-length protein sequence of the FOXP2 gene, alongside a GFP marker. So this will increase the expression of FOXP2 in the brains of these bats. And alongside this, as a control, we have a virus without the FOXP2 sequence, but that does carry GFP. We've injected these viruses into the brains of bats, and on this slide you can see the brain. We inject in one hemisphere the control virus, and in the other hemisphere the knock-in virus. So if we look at the control hemisphere and zoom in on this region, you can see that there are a lot of neurons in this part of the striatum, indicated by the blue nuclear stain. If we look at FOXP2 expression, we can see that it's fairly weak and sporadic. But if we look at the other hemisphere of the brain that's received the viral knock-in construct, we have similar amounts of neurons, but a much increased expression of FOXP2. And this can be quantified both looking at the intensity of FOXP2 across the tissue, but also the number of FOXP2 positive cells. And by both metrics, we see a dramatic increase in FOXP2 expression. So we've shown that we can successfully manipulate the expression of FOXP2 in a living bat brain. And this within animal design also allows us a very controlled way to look at the molecular consequences of this manipulation. Over multiple animals, we inject control into one hemisphere and knock-in into the other hemisphere, harvest the tissue, and use RNA sequencing to look at the effects on gene expression. We see that many genes are increased in their expression in the knock-in hemisphere, but about twice as many genes are downregulated in response to FOXP2 overexpression. And this is in line with the known role of FOXP2 as a transcription factor and its role commonly to downregulate gene expression. So with these transgenic models, we can now not just look at the molecular consequences of upregulating a gene like FOXP2, but also the effects on neural circuitry and vocal learning behavior. In parallel with this, we're now performing cross-species phylogenetic analyses to understand not just what's special about bats in the genome, but what's special about vocal learning species, looking at changes in the sequence of the genome, but also going beyond this to look at what's different in vocal learners about the isoforms of genes that are expressed, or the changes in protein structure or function, and also looking at pathways as a whole to understand if there's specific ways that pathways are expressed or activated in vocal learning animals. And in this way, getting a sense at a genomic level and at a neurobiological level of the underpinnings of vocal learning. So to finish there, I'd like to thank the members of my research group who performed these studies, in particular, Paolo, Stephen, and Janine, my funding agencies, and my collaborators, especially those in BAT1K, and thank you for your attention.
Hi, everybody. I hope you enjoyed this session as much as I had. It is just amazing to see the things that we're actually being able to do with these genomes. And I want to again thank all of our speakers. And just to remind everybody, if you've got questions to ask, make sure that you type in your name into the Slido, which is underneath the actual our talks from the platform, because we have some cool questions to ask our speakers. So I'm going to ask these questions in order of people pushing it up. So the first question is to Ariadna. Cool talk, thank you. How will you do the experimental validation of the candidate genes? And this is always something that we're asked.
Yeah, well, I'm not planning to do it. One of the strengths of BAT1K is that this big network of researchers with different strengths. So some of our collaborators, specifically Aaron Irvine in Shanghai, has developed a cell line of rhinolophids. Thomas Svanka also has developed a protocol for pluripotent stem cells with bats, and they will take care of those details for the experimental validation.
Yeah, and I think that's something that we all need to think about moving forward, is really how do we validate our in silico genomic hypotheses? But we're moving forward on that. So next question I have, again, it's to Ariadne, it's another one for you. So are the genes inhibiting the viral replication in the Hypsidaridae and the Rhinolophidae, are they SARS-CoV specific, or is it for any virus?
Well, they are for any virus, those genes that are under selection in the ancestral branches, because the divergence probably happened millions of years ago. And we don't know if coronaviruses were interacting with these bats at the time of divergence. However, those genes are under selection in specific terminal branches, meaning the species could have direct link with SARS-CoV-2. However, those in ancestral branches could be applied for viruses in general.
And the interesting thing is when you look at the diversity of the ACE2 receptor in bats, in the rhinolophids and the Hypsidaridae, it's so very different. So it does seem to be maybe an arms race going on between these lineages and some of these viruses. So yeah, hopefully our genomes can tell us a bit more. So next question we're gonna ask is to Veronica. So Veronica, great project. Could you talk a bit about the value of reference quality genomes for your project? How do these high quality genomes provide added value to the type of population studies you're planning? Always a question we're asked. Should we invest in the reference quality genomes?
Yeah, so always the answer is yes. So it's good to have really good reference genomes, because it really helps with the bioinformatics, like SNP calling and variant calling in general, but also if you're doing any kind of transcriptomics like RNA sequencing or working with methylation, it helps to have really good genome and annotation in order to know which genes are expressed and which are methylated and so on. So it really helps with the genomic projects or molecular projects that you're doing.
So you think that one good reference quality genome is worth it to be able to really advance and understand, I suppose, GWAS of conservation?
Yeah, for sure.
Okay, good. We all agree on that point, but I know it's an open conversation usually in this meeting. So I have a question now for Alexis. So Alexis, do you see less use of tryptophan in the other sugar feeding lineages that would have had a more carnivorous ancestor, such as your hummingbirds?
So that's a very interesting question. I'm afraid I don't really have the answer to that yet, but I want to kind of take the opportunity to point a difference with the bats. There is evidence that they changed a bit their metabolism ancestrally to prefer digesting lipids and proteins instead of using their hepatic glycogen reserves. So I don't know really if that's the case in birds, but that could be another reason why they would have such pressures to lose this pathway in contrast to other sugar feeders, especially given that this pathway is not easy to lose. It has a lot of, it will have a lot of adverse effects, especially on socialization and behavior and so on, because it takes away tryptophan from the serotonin catabolic pathway. Anyway, it's a very interesting question, ultimately.
And actually, Alex, there's another question for you that's just come in. So Alex, are the same molecular mechanisms, these premature stop codons, are these what is responsible? Is this the conversions that you see between the two back clades?
Oh, are they, if I understand the question correctly, is it the same exact mutations that we see convergently? No, because ultimately there is tons of ways you can lose a gene, and the point is to lose a gene, not exactly the exact same mutations. However, it's not excluded that we can see for specific transcripts, when we have more than one transcript, a specific exon loss. That's not excluded. And this is not something we have looked at in this case.
Okay. And I'm going to ask a question myself just to Sonia. So Sonia, it's cool stuff that you're working on with the vocal learning in the bats. Are you finding there's convergence with other vocal learning species?
We are in the process of asking that question. So we expect that there should be some convergence, because we do see some convergence with the work that's been going on in birds, so which are obviously much further away. So another reason we focus on the FOXP2 gene. But my hypothesis is we might see some things that are more similar in mammals that maybe don't go all the way across the birds and the mammals, that some things that are the same, some that are different. Hopefully next year I'll be able to answer that question.
That's a good answer. And then we have one other question here. So for Veronica, for your methylation bisulfite sequencing, are you going to use more than one individual? And if so, what tissues are you going to look at?
Yeah, so I will use more than one individual. So I have I will aim 10 individual per population. And the tissue is actually a really good question, because you might see tissue specific methylation, like you have RNA expression. Mm-hmm. So there you have to think about that, but you also need to be you cannot kill the bat. So you have to take samples that won't hurt the bats too much. So then it would be the wing tissue and blood. And especially blood would be really good, because there is the immune function that can be linked to urbanization, which could be interesting to see.
And that would be pretty cool. So I'm going to just check to see, do we have any more questions? Right now, guys, it's your last chance to ask questions to our panel. Does any of our panel members want to ask questions? Well, we're actually just 1 minute over time. So I would like to say thank you to everybody for taking the time. Thank you to everybody who's watching. Sonia, I'm sure you have some words that you might want to say to everybody too before we leave.
Similarly, thank you for the speakers giving great talks. Thanks for everyone joining us. Go to bat1k.com, join us at our next Batellite on the 5th of October, and join the community so you can answer all sorts of different questions with these genomes.
Okay, so thanks everybody.